Jai Hind everyone, I am Pankaj Kumar. Let's begin with the today's lecture. It is about the nucleotide and the nucleic acid. This is the series of the lecture about the nucleotide and nucleic acid in which we'll see its structure and its function. Metabolism we'll see in the next series, but here we'll cover in upcoming videos we'll cover the nucleotide and nucleic acid. So first lecture is about the basics of the nucleotides. So where you can see the nucleotide is everywhere in your body, and it's not only as a important in the form of the DNA or in the RNA, but it can be used as a form of the ATP, GTP as an energy molecule, or it can be used as a CGMP or CAMP as a signaling molecule. Okay, so it is having variety of function. Either it can will act as a cofactor for the enzymes like NAD, FAD, and it will be the intermediate metabolic it will be the intermediate of the metabolic reactions. So DNA and RNA major difference you know, but let's begin with the structure. What makes it different? So nucleotide consists of what? Nitrogenous bases and pentose and phosphorus. So if you'll take phosphate, nitrogenous base and pentose, then it will called as a nucleotide. If you'll remove this phosphate from the nucleotide, then it will called as a nucleoside. Nucleoside or nucleotide, it contain a nitrogenous base, and nitrogenous base basically is derived from the either pyrimidine or purine. So purine or pyrimidine, the important thing is pyrimidine and purine will be of heterocyclic. This is the basic structure of the nucleotide. You can see once pentose sugar is there, when either purine or pyrimidine as a nitrogenous base will be there, and phosphorus will be there. If you will remove this phosphorus, then this pentose sugar and the purine will called as a nucleoside, and if you will add the phosphorus, then complete structure will called as a nucleotide. This is the pyrimidine, which is there is only one cyclic ring, and purine having a two cyclic ring. So pyrimidine counting is very important. Nitrogen first, second position, third position, fourth position, fifth, sixth, and here we started from this nitrogen one, then six, so 1, 2, 3, 4, 5, 6, 7, 8, 9. In purine, we'll go for the anti-clockwise, and pyrimidine will go for the clockwise counting. This counting is very important. Why it is important? Because if you see the binding of the pentose, here if you see the binding of the pentose with the phosphorus, or you will see the pentose first carbon is binding with the purine. So if it is a purine, then it will bind with the first nitrogen. If it is if it is a pyrimidine, it will bind with the first nitrogen. If it is a purine, it will bind with the ninth nitrogen of the pyrimidine of the purine. So it is bind with the glycosidic bond. You can see if it is a purine, it is binding with the ninth position of the nitrogen, and if it is a pyrimidine, it is binding with the first position of the nitrogen. So while making a glycosidic bond, it will release the water and it will finally give the one glycosidic bond. What is the purine and pyrimidine? Purine contain the adenine and guanine, and pyrimidine contain cytosine, thymine in DNA and uracil in RNA. So their name will be very. It is you remember this is going anti-clockwise. 1, 2, 3, 4, 5, 6. 6 amino purine, and this will be 1, 2, 2 amino purine. Finally, we'll find there will be different name for this all structure. If you see the nucleotide and nucleoside will be for the adenine or guanine or for the cytosine, thymine, and uracil. The name will be what? If it is a nucleoside, the phosphate will be not there, then it will called as a adenosine. And if it is in the RNA, then it will called sorry, it will be in the DNA, then it is called as a deoxyadenosine. Same for the guanosine. If it is in the guanosine for the RNA, then DNA it will call as a deoxyguanosine. And in case of the cytosine, cysteine for the RNA, deoxycysteine for the DNA, thymidine and deoxythymidine. Important thing is that thymidine or deoxythymidine is present only in the DNA, so it cannot called you. The term is not used in case of the RNA. So uridine is not present in the DNA, so we'll use for the RNAs only. Like that, if you'll see the nucleotide, so adenine will call as a adenylate in case of RNA, and in DNA it will call as a deoxyadenylate. Same for the guanine, guanine guanylate, deoxyguanylate, s- cystidylate, and deoxycystidylate. Again, thymine is not present in the RNA, so it will called thymidylate or deoxythymidylate. Same. And in uracil called as a uridylate. DNA will have the ribose sugar will have a five membered ring that is called as a beta furanose. Five member ring is called as a beta furanose. In this, all five members will be not in the one plane. Usually, the two or three carbon, two prime or three carbon prime, will be out of the surface, out of the plane. So you can see either it is a C3 Indo puckering or C3 exo puckering. So if this 4, 1 or 2 and this red ball is in one plane, it means it is out inside of this plane, then it is called C3 Indo. It can be outside in the plane, then it is called C3 exo. Same for the second carbon. If it can be, if not 3, then which second carbon will go out of the plane or into the like inside the plane or outside the plane, but not in the plane. So this is called as a puckering. And remember, this prime we are using only for the ribose sugar atoms, but not for the purine. Now these are the structure as we seen here, and there is a monophosphate and adenylate, adenosine, guanosine, uridine, cysteine, which we already discussed. The structure you can pause and you can see for a while. Okay. Mostly the DNA is methylated in case of the human being, not only human being. In case of the plant and animals, the DNA will be methylated, but in case of the uh, infected cell, viral infected cell or the viral DNA will be hydroxymethylated or glycosylated. This is the important characters of the DNA methylation. And as well as you can see the tRNA having a inosine instead of adenosine. Okay, so inosine will replace the adenosine, or you can say inosine is a different form of adenosine. But what nitrogenous base it contain? It does it contain not adenosine, uh, adenine. It contain, instead of adenine it contain hypoxanthine. So like that, pseudouridine will be there. Pseudouridine having a uracil which is attached with the fifth carbon instead of the instead of attaching with the first nitrogen. As you've seen, the purine will be sorry, pyrimidine will attach with the first nitrogen, first position of the nitrogen, and if it is forming a pseudouridine, it is attaching the ribose sugar with the fifth position carbon. So puckering you seen, and we'll see next their nature. So phosphate is there in the DNA, and if it is the phosphate is there in the DNA, the charges will be there negative at neutral pH. The DNA have a negative charge, but what about the individual purine or pyrimidine? So individual purine and pyrimidine will not have any charge at a neutral pH. They are hydrophobic, so it is insoluble. Only purine and pyrimidine, but it can get the charge in the different pH. It will induce a charge into different pH. So purine and pyrimidine can induce a charge, but at neutral pH, purine and pyrimidine are hydrophobic. But DNA, complete DNA or nucleotide with phosphate, it is negatively charged at neutral pH. So in upcoming videos, we will see the will see the mm, more about the structure of the DNA, and the DNA doesn't have the OH at the second prime, but RNA have the OH at the second prime. This is the known thing. But one more thing is left, the tautomers. So different form of the uracil will be formed, or adenine or purine will be there, and that is called as a tautomer. Okay. So tautomer will be present in this form. I will try to show you here. You can see lactame, and double lactame form is of the tautomer of the uracil. So at a, depending on the pH, they will have a different tautomer, different form of the structure. So this is called as a tautomer. You can see lactame, double lactame. So these are all the tautomer of the uracil. So their absorbance will be at 260 nanometer UV spectroscopy. You will see their absorbance maximum at 260 nanometer, and the highest absorbance you will find in the AMP, then GMP, then UMP, then CMP, and TMP. So these are the absorbance graph. So hope you enjoyed this video. We'll meet in the next class, and if you want any uh, topic, then you can ask me there in the chat box. So till next video, Jai Hind. We'll meet in the next video again.